Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. For this first one, it says, which statement describes the properties or functions of cholesterol? So cholesterol, remember, is a type of sterol. So it's a steroid alcohol, and it's another type of lipid that's found in cells. And its role is to stabilize the cell membrane and make it less fluid. And it can also be used in making hormones and other kind of things like that. It's like steroid hormones. So first of all, increases the fluidity at high temperatures? Nope. It is an unsaturated fatty acid because it contains carbon-carbon double bonds. So remember that fatty acids have can be unsaturated because of the carbon-carbon double bonds, but that doesn't make it a fatty acid because a fatty acid, remember, has your sort of long what's the word? Your fatty acids have those long acid groups and those long carbon chains as well. So that's not what is going on here because a fatty acid, remember, has an HO, C double bond O, and whatever the hell it is on this side. And that's not what cholesterol has as in its structure, so that one's out. It's used to make hormones, yes, and it's very hydrophilic, so it's attached to fatty acid tails in the membrane. No, because remember that the tails are hydrophobic, and so that one's out. Which statement best describes triglycerides? They're broken down by condensation reactions. No, it's hydrolysis. They are polymers of glycerol and fatty acids. Remember, it's not a polymer because you can't have a repeating unit. You've just got glycerol and 3 fatty acids, so nope. They contain ester bonds? Yes. And glycosidic bonds is between glucose. Alright, so Kendall mint cake is made from sugar, flavored with peppermint, and was famously taken on the 1953 ascent of Mount Everest. Which statement describes why climbers now prefer to take chocolate? So chocolate contains fat. Fat stores and releases less energy per gram than sugar. No, because you've got like because it's all condensed and it's all kind of put into these lipids and stuff. So actually, it's a storage molecule. So surely you'd want more energy per gram than sugar. So no, fat. Chocolate contains fat. Fat stores and releases energy more than sugar. Yes. It wouldn't make sense because surely if it was less energy per gram than sugar, they just took the sugar in itself, or like something with just pure sugar. So it's that. Next, it says the graph below shows the effect of changing substrate concentration on the rate of an enzyme-controlled reaction. So as the rate of reaction, sorry, so as the substrate concentration goes up, the rate of reaction climbs up, but you can see it levels off here. So just have a quick think why. The rate is shooting up because you've got more and more substrate colliding with those enzymes, forming enzyme substrate complexes. But over here, there's so much substrate that all the enzyme active sites are filled up. So they can't do anything. So it's saturated and it levels off like that. So then it says, which graph shows how increasing the concentration of enzyme would change this reaction? So again, so it's got to be whichever one just basically. So think about it this way. We're infinitely increasing the substrate concentration. If we make the enzyme, if we had more enzymes, let's say we had like, I don't know, 25% more, it would go up, it would up and up, but then eventually they would all still saturate too. So you would have the leveling off just higher up. So it's not that one, because that's leveling off lower down. So that's actually taking enzymes out or inhibiting them somehow. That's unchanged. That doesn't make sense, because again, we've got more enzymes, so it should level off at a higher rate. So nope. C is the only one it can be. And D, well, it would level off at it would level off at the same height, because it would increase the same, just a higher rate and level off like that. So it wouldn't make sense for it to be drawn out like this. So it's C. Number 5 then. It says, which statement about the effective temperature on enzyme substrate controlled reactions is not correct? So at temperatures above 40 degrees C, the rate of all enzyme-controlled reactions decreases. Well, that's not correct because we have enzymes that can work at way higher things. For example, in PCR, DNA polymerase A, already off the bat. Increasing the temperature above the optimum changes the tertiary structure of the active site. Not well, it does, to be honest. So yes, increasing the temperature increase the kinetic energy. Well, that's true. And increasing the temperature increases the probability of enzyme substrate collisions. That's true. So it has to still be A. Next, circulation of the lungs involves muscles working together. Which one describes expiration? So in expiration at rest, the external intercostal muscles and the diaphragm relax, and the rib cage moves down and in. So basically, the way I remember it is that when you're breathing in, your diaphragm is the muscle. So if I got these really poorly drawn lungs, the diaphragm is here. It flattens and goes down, and it kind of like drags the lungs down with it as well because of the negative pressure in it. So it makes those lungs get bigger. And also, those external intercostals contract, and it makes your chest get bigger. So if your chest goes big in, when you're breathing in, the volume increases and the pressure decreases. And when these two tend to be inverse. When the pressure decreases, things like to move down from a high to a low pressure. So the pressure is so low inside you that it's actually higher on the outside than in the air around you. So it drags air in. When you're breathing out, all you're doing is you're just doing the opposite of everything that you did at the start. You're just relaxing the muscles. Remember, muscles contract and they relax. You're not actively pushing the air out, or you would be exhausted. Imagine trying to breathe every single second of the day and you're breathing in forcibly and out forcibly. You would feel exhausted. So it's actually just a passive process where all it does is the external and diaphragm relax, the opposite. If you're forcibly breathing out, that's when the internal intercostals kick in. But for the most part, when you're just doing it at rest, it's just a relaxing. So when you're breathing out, think about what's happening. Your rib cage is going down and in. It's increasing the pressure inside you, and it shoves the air out. So the only one it can be is A, which is not a component of a chloroplast. So Christie is found in mitochondria. Bang, A. All the rest of them are. Remember that these chloroplasts can make their own little proteins inside them as well, so they do have ribosomes in them. The following information is needed to answer questions 8 and 9. So Glass comparing the number of chloroplasts in the leaves taken from plants of the same species going in two areas. One had a high light density and the other one had a low. One student examined a cross-section of a leaf under a microscope and they counted the number of chloroplasts in each cell in each field of view, and the results are shown in the table. Which one gives you the correct value of the mean, median, and mode? So the mean is just all of them added up divided by the number that you have. The median is the middle number, so you're going to have to write them in order from smallest to biggest. And then the mode is the one that just appears the most. Just to save time, I'm just going to go ahead and say that's C. So which of these options is correct? The correct procedure for statistical analysis of the data collected by this class. So we need to look at this and think what they're doing. So they're looking at the number of chloroplasts in different cells, and they're comparing it between high light intensity and low light intensity. So what they're going to be doing is they're going to compare the mean numbers of them. It wouldn't make sense to compare any correlation stuff because nothing is correlating with something else. It's not like one's going up and the other one's going down, because that's when you're changing one variable and the other variable changes. And it wouldn't be a chi-squared test because we're not looking at expected versus observed. So, for example, I'd expect that 1/4 of all the offspring of this cross in a pea plant would be, small and wrinkled, for example, but it isn't. So the only one that it is because you're comparing the means between two different groups. It has to be the t-test. So now we need to look at which one of those fits the t-test bill. So calculate the mean number of chloroplasts high and low intensities and then use a paired t-test. So it wouldn't be paired because paired is when you're using the same group and then doing them again. So it's out. It's going to be unpaired. Paired would be, for example, if you had people exercising. So you measure their heart rate before they exercise and the heart rate after, and then you work out the mean heart rate before and the mean heart rate after. So it's the same thing, just before and after, but you're comparing the means. T-tests are all about means. If it's unpaired, what it means is you have one versus the other. So the heart rate in one group versus the heart rate of another group, completely unrelated, not the same one. Next, 10 says, which is an example of cell signaling. So cell signaling is all about, something being released from one cell or something like that, binding to another receptor and then triggering something else in that other cell. So binding of a virus to a receptor on a cell surface membrane in an epithelial cell will know because viruses aren't cells. Next, binding of acetylcholine to cell surface receptors and sodium ion channels in the postsynaptic neuron. Well, yes, because that gets released from the presynaptic membrane and so forth. So that one's got to be it. Let's have a look at the other ones. Co-transport of glucose and Na plus ions across the cell surface membrane. Well, that's not cell signaling because it binds to it and moves in. It's just movement, so no. Operation of the Na plus K plus pump in the cell surface membrane. Again, it's not signaling. It's not making another cell do something as a result of things binding. So it's next. It says which of these statements describes an aspect of homeostasis. So remember that homeostasis is all about something goes. So you had a stimulus, which is where something goes a bit whack, and it may be that it's gone either too high or too low, and then you have a whole load of responses till effectors eventually bring it either back down to the normal or back up to the normal, and it just keeps oscillating between that. So it's all about keeping a stable internal environment. So let's see which one of these would be it. So effectors detect the change and cause a release of hormones that bind to target cells. No, effectors don't detect the change. They bring about the change. It's receptors that do. So that one's out. Homeostasis maintains the internal body temperature at a constant level. B, yeah, because it does. It keeps it at normal. Homeostasis involves positive and negative feedback. Not necessarily, because it can be negative feedback on its own. So no. For example, if you have like insulin, if you're looking at just insulin action, that only brings things down. There is a glucagon brings it up. That's still a negative feedback mechanism, even if one is going down and up. The reason being is because, again, you're changing it to the normal, right? So that's negative feedback. So too high, back to normal. Too low, back to normal. Positive feedback is bang, shoots off. So you can't have positive feedback in homeostasis because you're actively taking it away from the normal. So that one's out. So, and then it says receptors coordinate nervous and hormonal responses. No, they don't. So glands would release those hormones, and then nerves would coordinate those nervous responses in the central nervous system, so it's got to be B. Next, it says linoleic acid. Linoleic acid is a fatty acid found in sunflower oil. The equation for the oxidation is this. So it's C18H32O2 plus 25O2 gives you 18CO2 and 16H2O. Remember that the respiratory quotient is going to equal the CO2 produced over the the O2, consumed. So that's going to equal 18 over 25. If you do that, 18 over 25 gives you an answer of 0.72, which is B. Oh, wait, sorry, B. Why am I ticking these? Sorry, ignore that I've been ticking that. I didn't realize. Whoops, it's B. I'm on autopilot. Sorry, whoops. Next, an electromyogram (EMG) records the muscle electrical activity, and a figure shows the AMG trace of a muscle undergoing fatigue. Which of the statements about muscle fatigue is correct or are correct? So if we look at all of them together, increased acidity in the muscles during long periods of contraction is a cause of muscle fatigue. That is a possibility, yes. Fine. There is a decrease in the frequency of the signal that the EMG traces the muscle becomes fatigued. No, because you can see that they're still, equally frequent. It's the amplitude is dropping. The size of them is dropping. So that one's out. Next, it says an increase in amplitude. Well, no, because you can see it's again going smaller. The frequency is how many times this happens. The amplitude is how tall they are. So the amplitude is actually decreasing, so it's just D. Next. Which of the statements about the relationship between photosynthesis and aerobic respiration in plants are correct? So let's have a look. During the day, photosynthesis produces all the ATP in the cells that they require. Not really, no, because they still will respire. Respiration never stops, so that's out. And also, it produces very little ATP, and that ATP is used to go into the Calvin cycle to do its job, so not massively, no. CO2 produced in respiration can be used as a carbon source of photosynthesis. That is correct. Oxygen produced in photosynthesis can be used in aerobic respiration. Yes. So it has to be only 2 and 3, which would be C. Next, it says erythrocytes are formed from bone marrow stem cells. During this process, they lose most of their organelles. Which statement about respiration in erythrocytes is correct? So oxygen bound to hemoglobin is used by erythrocytes in aerobic respiration. No, that's the whole point of it is to carry it around. So no. They do not respire because their cell surface membrane is impermeable to glucose. No, because if that were the case, they would not exist. Next, they respire aerobically because they always have adequate supplies of oxygen. No, because they don't have any organelles. Next, they respire anaerobically because they do not have mitochondria. Yes, because they don't have any organelles. They've told you in the question that as well, and they don't. So now it says dicotyledons are a large and diverse group of multicellular plants that include oak trees and dandelions. Explain using examples of transported substances why multicellular plants need transport systems. So think about it. The question's asking you, with examples, explain why they need these transport systems. So in plants, they're using mass transport and stuff, right? Now think about what the alternative is. The alternative is that they've got to, like a prokaryotic cell, wait for everything to come in and diffuse in. Now, if you've got this big ass tree, things aren't going to be able to diffuse into the middle. So a lot of this about mass transport is talking about maximizing diffusion. So you need to think what factors affect diffusion. So your options are temperature, not relevant here, doesn't matter. Your concentration gradient, diffusion distance, and surface area to volume ratio. So first of all, the diffusion distance would be too large if relying on the right diff for short, diff in the outer surface, because other smaller cells can just diffuse things in and out of the cell surface membrane. How the hell are we going to get into the middle? It's going to take bloody ages. And as well as that, let's say for example it was just that they went straight from they just from the water they picked up from the roots, they're just waiting for osmosis to carry it across the entire plant and not using a xylem or phloem. That would take ages. So they are large, and also diffusion would take too long over long distances. Think of it this way: if we didn't have blood in our circulation, you're waiting for oxygen to eventually get to the muscles. It would take absolutely ages. Another option is obviously low surface area to volume ratio, and then we need to give examples, of which I've already given some. So water from roots to other parts of the plant, and then as well as that, if we're looking at large diffusion distances or something, for example, we could be talking about let me think. Well, I guess we could talk about we've already used the water example, so we could talk about things like ions, for example, or we could talk about sucrose from the source to the sink via the phloem. Because if we just kind of waited on these to diffuse over time, it would take absolutely ages. So we have those in place to do that. So it's about thinking about the factors that affect this process and why we have these to adapt to making that process better. Next, it says figure 16.1 is a light micrograph of a transverse section. So it's a slice like that, this bit here, and you're facing it here, through the leaf of a dicotyledon. Next, it says identify the figure B in figure 16.1. So the inner ones are the xylem, so the outer one's got to be phloem. It says state two functions of the the structure A. So we know that's xylem. So the whole point of one of them is the obvious, which is transport transport of water. And then you might well also want to ask, why does the water matter? It also provides structural support too, because remember that like water and stuff provides turgor pressure and keeps plants turgid. It helps them stand up. So that's also another important reason it exists. Figure 16.2 on the insert is a colored scanning electron microscope of a longitudinal section through the vascular tissue of the stem of a dicotyledon. Explain how two features visible in this show that it is adapted to its function. So you can see here that is, you should be able to recognize that those are spirals of lignin in the lumen of the xylem. So you've got lignin in the xylem. So then you need to obviously explain why it's important. So what it does is it prevents collapse and keeps the lumen open. That spiral prevents it from sort of the walls sticking to each other and it keeps it open. So there's a big one. What else have we got? So also remember that we've got these non-lignified pits as well, or bordered bits. So why are they useful? Well, it allows passage of water, between the xylem or like in and out of the xylem. Remember that these spirals of these lignin, what they do is they just kind of help keep the structure there, but they don't actually transport water itself, but those empty gaps allow water to move freely. So now it says a mycorrhiza is a mutualistic association between a fungus and a plant. The fungus penetrates root cells and assists with the uptake of water and nutrients. A group of scientists investigate the role of mycorrhiza in the uptake of phosphates by plants. Wild-type barley plants produce root hairs. Mutant BRB barley plants do not produce root hairs. So wild have root hairs, BRB have no root hairs. Right? So then it says BRB mutant and wild type plants are grown in pots, and half of them had mycorrhiza. So half of them had mycorrhiza added. So myco, no myco, myco, no myco. After 28 days, what they did is they measured the dry mass and the phosphate content of all of these things going on here. Fine. And the results are shown in the table. So the plant type, so again, just look at the table and figure out what's going on first before you jump in. So you've got BRB mutant. It says mycorrhiza fungus added, no, yes. So obviously we're comparing without and with mycorrhiza, and then with and without mycorrhiza in the wild type. So when you're doing these kinds of things, just describe to yourself what's going on in the table. Start off by just comparing like for like. So we're going to compare the mutants. We're going to see what happens if the mycorrhiza is added or not. So the dry mass, if we then look at between them as well, you can try and compare the differences. So mutant versus wild, the dry mass of the mutant is lower than in wild with no mycorrhiza, and then mutant versus wild, when they're at the phosphate content, it's lower as well. So wild types, mutant versus mutant, tend to have more mass and dry mass and phosphate. So if you compare the BRB mutant with mycorrhiza and wild without mycorrhiza, the gap gets a bit narrower. So they start to get closer together. Fine. So then it says the scientists concluded that mycorrhiza compensated for the absence of root hairs in the mutant barley BRB plants. Evaluate this conclusion. So first of all, figure out what the actual statement is. So they're saying mycorrhiza increases root hair, absorption in BRB plants. So that means you can't just go ahead and jump into saying, well, they only did it in these plants, because they're not concluding that, it's their conclusion is only on this experiment. They're not going ahead and saying every plant in the world will do the same. So when you're doing this, try and think of, when you're evaluating it, you're going to say what supports it and what goes against it. So if we look at what supports it, it's, first of all, we said, like I just described there, adding mycorrhiza increases dry and phosphate mass in the mutants, which supports the fact that this may help them increase absorption in the roots, because these mutants don't have root hairs. All right? So then you could suggest what could be going on. Well, maybe the fungus could stimulate root hair growth. How was I able to conclude that? Well, they've told you in the question that it's the role of mycorrhiza in the uptake of phosphates by plants, and they've told you that, what else? These fungus penetrates the root cells and helps them do their job. So if these lot don't have root hair cells, these mycorrhiza can't actually penetrate them and do that. So there's got to be some root cells coming into play here for this to work. So it could be that they're actually making the root cells grow so that the mycorrhiza can get in there. So you've got to use what's in the text, right? So now let's look at what's against it, or not against, but just limitations as well. So if we think about it, well, in the wild type, adding mycorrhiza actually decreases dry and phosphate, and as well. So we can adding it actually decreases it. So because the mycorrhiza attaches to the roots, surely if they're making it grow and they're compensating for root hair problems, then it would make it go higher because these have roots. So maybe it'd make them work more. So that actually kind of goes against what they've just said. As well as that, well, let's see what else is going on here. Um, well, if you look here, we don't have any controls. We've just been given mycorrhiza, no mycorrhiza, mutant mycorrhiza, no mycorrhiza. We don't have any idea on like what the no mycorrhiza groups had. So we don't know if there was anything like else added, whether they used the exact same stuff, just took the mycorrhiza out, or whether they put like I don't know beads or something in to simulate having mycorrhiza. So it's things like that you've got to think about because the experiment doesn't tell us that. So no info about control. As well as that, we actually don't know anything about sample size too. All we know is that they just got half. So there's no. Whoops, why is it rainbow? Okay, cool, no problem with that. Sample size. Because again, I'm looking at this experiment and critiquing it like that. What else do we know? We know that there's these SDs here, but we don't actually, or like there's sort of overlaps and stuff, but there's no actual p-values or statistical tests. So, like I said before, to be honest, admittedly, I didn't read the whole question. There's no overlap, which again supports that. Like, it could be that there's no overlap, but we haven't done a statistical test to check that this not overlap is not due to chance or not. So we don't actually know if these non-overlaps, sorry, I should say, are actually true or not. So it says the thermal properties of water allow organisms to live in an environment with relatively small changes in temperature. These properties of water also make it efficient as a coolant, e.g., in sweating, or by absorbing large amounts of heat. So it says outline how other properties of water are essential for sustaining life on Earth. So when it's talking about that, it's saying why is water important and describe its purposes and how these lead to it. So first of all, we need to talk about its properties. So its chemical properties, structure your answer, right? So it's a polar molecule, which gives it all of its funky stuff, right? And the reason is because there is a slightly or delta positive charge on the hydrogen atom and a delta negative charge on the oxygen atom. And this results in hydrogen bonds forming, and that's what gives water its funky properties. All right? So what happens is that large numbers of these are strong as well, and little numbers of them are quite weak. So as a result, let's talk about these are what give it its physical properties. Its physical properties are, well, first of all, one of the funkiest ones is that when you sort of cool this down, the hydrogen bonds actually spread out more. So that gives you ice gives you a lower density than water, so it floats. So then you need to think, how does that mean it's essential for sustaining life on Earth? Well, if a river, like for example, has an ice layer on top of it, the ice can actually insulate heat below. So it floats. So, e.g., if like the river top freezes, ice insulates, below, and that actually can keep life sustained. What other things are there that water can do? Well, because of its properties with regards to, like, for example, its hydrogen bonds and all of that kind of stuff, it actually allows for cohesion as well. So cohesion tension. So you could talk about that and how it gives you your sort of column of water in transpiration, and that means that water, it moves through a xylem from roots to leaves in a whole column, which is important because it means water is needed for photosynthesis and so forth. Like, if you couldn't have that, then you couldn't actually force the water to go all the way. It gives the plant that circulation. Other properties that make water important is that it's a solvent, so it can act as a medium for chemical reactions. Because, like, when enzymes and substrates collide, they have to be dissolved. They have to be able to freely move. You can't just have a solid block of solid doing stuff. So that's an important thing. And it also means you can use it for transporting stuff too, as well. What else can we use? Well, not just that. There's other things too. So, for example, it's used in hydrolysis, which is important because then you can use it to get, like, for example, get glucose out of substances, or you can use it for getting rid of waste products as well. So that's quite important. And as well as that, while we're here, you can also transport waste products out as well. So that's a couple of things you can mention. Other stuff as well is it can be used as a lubricant. So for example, in your lungs, you have the pleural fluid lining the pleura. It means that it doesn't stick to itself and you can like actually breathe in and out. Otherwise, it would just kind of either be stuck to the end, and if you like, it would just rub against each other and it'd hurt and be not great. So that's another thing as well. And also, if you really want to reach and as well, you can. It allows water to move. Like, sorry, it allows light to go through it. It penetrates it. So in like aquatic organisms living at the bottom that need light, it can still pass through to the bottom. So those are a couple of them. It's just basically essentially saying here's its chemical properties. As a result, it leads to all these funky physical properties, and these are important for sustaining life on Earth. So you got to structure your answer. So now it says a student investigated osmosis in plant cells. They used cylinders of potatoes cut with a cork borer and measured the change in length of the cylinders after they were placed in a sucrose solution and left overnight. So they were given 100 centimeters. So I'm just going to write this down. So 100 centimeters cubed, and the concentration that they were given was 1 mole per decimeter cubed sucrose, and they wanted to make 30 centimeters cubed of each dilution. So now it says complete table 18.1 to show how the student should prepare those solutions. So if they're gonna, all of these want to be a final volume, and this is the final volume of concentration. So a quick thing you can do here is you can do C1V1 equals C2V2. So that means this is the concentration that you have. This is going to be how much of the original stuff you're going to use to make this. And this is the concentration that you want. This is the volume that you want. So if we rearrange that, V1 is the volume of the original solution you're going to use to make all of this, C2V2 over C1. So the volume for each of these would just be the concentration that you want. So for example, we pick the first one, 1.0 times the volume, which would be 30 over 1,000, over the original concentration that we had, which was 1 mole per decimeter cubed. So that's the volume of this stuff here that you can use. So 1 times 30 over 1,000 over 1 would be 0.03 decimeters cubed. But remember, just to get it back into centimeters, you times it by 1,000, so it would be 30. And then you can just switch it up, because now the next concentration we need is 0.8, which would give you 0.024, which is 24 centimeters cubed. And also I've got to put the decimal places, sorry. And then you can just carry on doing that. So if we keep doing that, it'll be 18, it'll be 12, it'll be 6, and then it'll be 0. And then it's the volume of distilled water to make it 30. So these are just how much water you need to add to get it up to this point. So 0, 6.0, and then what, 12, and then that's what, 18, and then it's 24, and then 30. So that's a quick way of doing it mathematically, or you could kind of just sort of work it out bit by bit, but I just like doing things. Now it says the solution given to the student was prepared using 34.23 grams of sucrose and 100 centimeters cubed of water, and the sucrose was measured on electric balance using the following procedure. So the mass of the weighing boat was this, the mass of the weighing boat plus sucrose was that. The balance records masses to 2 decimal places with an uncertainty of that. Calculate the percentage uncertainty in the mass of sucrose. So the percentage so remember that percentage uncertainty is going to be your uncertainty over the value that you've recorded. So they want it in the mass of the sucrose. It's going to be this. So in order to get this, they've used the scale twice. So that means the uncertainty in this thing is going to be 0.01 times 2, because they've read it with it empty and then with sucrose and subtracted it, divided by the thing that you actually got, which is 34.23, and then you're going to times it by 100 to turn that into a percentage. So if you do that, it's 0.01 times 2 over 34.23, and that gives you 5 times 10 to the minus 4, but I'm timesing it by 100, so I should get 0.058, which is roughly 0.06. Because we want to match the number of decimal places that they've given us as well. So now it says the students' results are shown here. So here's your concentration, and the concentration is going down. And you can look at the quick percentage changes here as well in the means. So this is all the numbers to actually do the calculating. So as the concentration drops, you can see that the percentage change in length goes up. Cool. So then it says one of the students' results was anomalous. So we've got to look at where it is. So it is in this column here, because like you can see, that's like a nice consistent sort of value there. These are all nicely and somewhat consistent here. Those are decently consistent, positive, and then just a negative out of nowhere. Like, hello, where's that come from? And you can see that those two are kind of close and positive, and that's like way out. So that's the one that it's going to be. So minus 1.50. Next it says state one variable that should be controlled and give a reason for your choice. So again, where we're looking at putting these potatoes in sucrose and then looking at how much water they either take up or lose. So think about the factors that affect osmosis. So it would be temperature. As well as that, we're putting potato cylinders in. We need to make sure we're using the same type of potato because lots of different potatoes have different water potentials. And also, you want to make sure that you have like the entire surface of this potato like cylinder covered. We don't want it to sit on the bottom because well, that surface is not in contact with water. So that's another thing you could say. So I'm just going to pick temperature, because it affects the rate of osmosis. But any one of those is fine. You could also say, yeah, those. Now it says the table puts structural and functional differences between the different types of muscles. Put a tick in each box which one applies to that one. The first one's been done for you. Skeletal muscles are multinucleate just because that's just how they are. It's a bit to do with how they form and stuff. Fibers are branched interconnected in cardiac muscle. So the reason that they are doing that is because they interconnect and they can simultaneously contract at once. Whereas in striated or skeletal muscle, they are regularly arranged. They're regular, nice fibers like that, so they actually contract just in like a single direction. Whereas like cardiac muscles, you don't really want it to do that. You want it to sort of get a simultaneous contraction at one go, bang. And as well as that, under conscious control. So I mean, it says involuntary in the title, so that one's out. Cardiac, you don't have to think about beating your heart, skeletal. So now it says identify 3 proteins that make the filaments labeled as Z. So they've told you that these are transmission electron microscopes of striated and muscle. Okay, so what we need to do is let's just have a quick look at this. So we've got striated muscles. So remember, striated muscles are made of lots of repeating units called sarcomeres. So sarcomeres are marked with 2 Z lines as well. So we've got a Z line here and a Z line here. So that whole thing is one sarcomere, right? So sarcomeres, remember, have your alternating light so your actin and your myosins and stuff like that. So where it's darker, you're going to have actin and myosin, and where you're going to have the lighter parts, it's just going to be actin on its own. The H zone is the bit in the middle where there's only myosin present, right? So now it says 3 proteins that make up the filaments labeled Z. So here is what they're looking at. So we're looking only at the light part. So the light part is made of actin only. There's no myosin here because it's light. There's in that picture. So they need to think, okay, well, what other proteins are in actin? So actin, remember, just as a really rubbish drawing, it has the actin-myosin binding site, but that's blocked by 2 things, troponin and tropomyosin. So that's what the other ones are. So tropomyosin is a protein, and troponin is attached to it as well, so that's another protein. So there you go. Can't be myosin because it's pointing specifically out there. Labels X and Y show 2 distinct bands. Explain why these bands are different. So if you look at X, you can see that it's darker. So X contains myosin, right? And then Y contains, myosin and actin, but like it's looking at the whole thing. So the key difference is that it's myosin, and then Y contains actin only. Next, use your knowledge of the sliding filament model of muscle contraction to explain why figure 19.2 represents contracted muscle. So when you're contracting muscle, the sarcomeres become shorter. So the Z lines that mark the edge of them actually shorten. So already off the bat, you can say that. Sarcomeres are shorter. And then if you think about it, if the sarcomeres are shorter, something has to be moving about. The things that move are the actins. Remember, the actins get dragged across by the power stroke in the myosin. So the myosin's going to sit in place. That's going to stay the same. But the actin's going to move. So what happens is that I band, or the light band, is smaller. But because remember, that myosin ain't going anywhere, it's chilling. The A, or the dark band, is the same. And remember that the H zone is just where there is only myosin. So if the H zone, if the actin's being squished together, you're going to have a narrower H zone. So H zone more narrow. So if I find a quick picture just off of Google real quick. So every single sarcomere has these points that mark the ends of them, the Z lines here like that, and these are repeated. So the Z, if they're contracting, these Z lines by definition have to be moving closer together. But remember, like I said before, that when you're doing the actual power stroke, the myosin head hinges that way and drags the actin across. So the actual myosin isn't going anywhere. So the myosin stays in the same spot. So the A band is the same because the A band is all the actin-y dark stuff, right? Because the sarcomeres are getting shorter and the actin's moving closer, these are going to narrow this part here because that's H zone here is only where there's myosin. And because now we're actually contracting, we're moving the actins together, the H zone becomes smaller. And I guess also you could say that there's more overlap between the actin and myosin as a result, but those are the points that I'd make. So now it says, after death, the concentration of ATP in muscle cells decreases. During this time, the muscles become rigid in a process called rigor mortis. So suggest why a lack of ATP leads to muscle rigidity. So you need to think of the role of ATP in muscle contraction. So when a muscle contracts, you've got your actin chilling here. Sorry, whoops. You've got your actin here, and you've got the actin myosin binding site here, and you've got your myosin head there. Right? The goal is to get that there and then swing it across like that to actually drag the actin this way. And that process to actually, do this stuff requires ATP to actually move the head, but also this thing needs to break off so that on the actin at the next point where the actin myosin binding site is, this myosin head can bind to it as well, and then drag it further like that. When you're returning to the rest stage, this actin myosin binding site breaks and then the actin slides back into its normal place. So if you have no ATP, no ATP means no breaking of the actin myosin cross bridge. So that means that the actin cannot slide back to normal position, so the muscle stays contracted. Because in summary, again, the myosin head binds here, drags it across, and then when you're back to rest, the myosin head detaches using ATP to break that bond, and the actin just slides back into place like that. For as long as it's staying slid in the other place, like as an out of position, you're contracted, so that's what causes that. So now it says muscle contraction needs energy in the form of ATP. Describe the different sources that are used in muscle contraction during strenuous exercise. So your sources of ATP, the ones that you can have, well, first of all, you've got your phosphocreatine system. So I'm going to write PCR for short. So your phosphocreatine system is where its ATP is made from creatine phosphate. The phosphate is it binds to an ADP plus P, ADP, oh, whoops, I'm so used to saying it automatically, to make your ATP, and that is in the muscle cell sarcoplasm. So that's one source of it, but the problem is that it runs out quickly, so it's good for short bursts of exercise. Right, now think of where else can you get ATP. There's only two other places. First of all, aerobic resp. So that happens so the main place where ATP is made is oxidative phosphorylation, and it requires O2. And then it says, well, we need to compare them during their processes as well. So this is the most ATP. However, it's limited by your O2. So if you've not got enough oxygen there, so it's better really for long, low intensity exercise like running or something like that. Then anaerobic is next. So this is good for strenuous exercise because it's pretty quick. All it does is it only requires glycolysis, but makes 2 ATP. So it's 2 ATP, so it's less, right? And then you need to talk about, well, can this carry on? Well, the NAD is regenerated, so the glycolysis can continue. But the issue with this though is that you can get a lactic acid buildup, so that's its limitation. So with all of these, you can see that I've talked about just the process, where it's coming from, and then what its limitation is. So in the first one, the PCR system's great, it's really fast, but the only issue is that it's only good for short bursts of exercise because it runs out very fast within seconds. Aerobic respiration's pretty good, but it requires O2 to be delivered there, and it's quite more long and slow, even though it gives you the most ATP. Anaerobic's pretty fast because all it requires is glycolysis, but the issue is that you can't do it indefinitely because you get a buildup of lactic acid. So now it says, Oplognathus insignis is a fish species that lives in salt water and may be suitable for use in fish farming. Scientists investigated the growth of O. insignis at different temperatures to help decide the optimum temperature for farming. And then we've got a respirometer here. The diagram shows a single fish, but the respirometer is large enough to hold many. So what's going on here? We've got a water supply, we've got a respirometer, we've got a fish chill in there, we've got some water out, we've got an oxygen probe and a measure and stuff like that. Fine. Suggest 2 modifications the researcher would need to make this apparatus investigate the effect of temperature on the rate of respiration. So if we're doing, what that means is if you're investigating the effect of temperature on the rate of respiration, you are going to change temperature and see how resp changes, right? Now, if you look at this diagram, I have absolutely no idea how you're going to change the temperature. There's no way of changing the temperature. So, first of all, you probably want to add like some sort of thermostat to control temperature. And then as well as that, a thermometer to actually see what the temperature is. to measure temperature. As well as that, I probably put the oxygen probe as well, another one like before, so I can compare what's happening before and after and see how much the fish is taking up. Next, it says, suggest one advantage of using a computer to collect data. So if we did it without a computer, what you'd have to do is you'd have to just measure it every 10 minutes and so forth, and then you might miss out gaps or something like that. So this would just continually collect data nonstop, whereas with a human, it might be like bits and bobs of it. So we actually miss out what's in between these. So it allows continual data collection, which means you actually, it allows that so you can like not risk moving, like missing out the in-betweeny bits. Next, it says a constant flow of water past the oxygen probe allows continuous monitoring of oxygen concentration. Explain one other reason why it's necessary to have a constant flow of water in the respirometer. Remember that we're looking at how temperature is affecting the rate of respiration. If we don't have constant oxygen, fish ain't going to have no oxygen left to respire. So they're not going to do it aerobically. So none of the oxygen is going to get mucked about with. And therefore, we can't actually investigate how temperature changes respiration. So it's to make sure that that fish is always aerobically respiring. So ensure fish is aerobically respiring. And how this by maintaining adequate O2, because if we don't give it enough oxygen, it might switch to bits of anaerobic respiration and that'll muck up our results. So we want to make sure that the oxygen levels and the type of respiration being done is controlled by that. Because then we know its temperature, looking at the rate of respiration that uses oxygen, because we're measuring oxygen. So we want to make sure it's always aerobically respiring. So we need to make sure it's got enough. As well as that, it also kill the fish. So I guess there's that too. Next, it says, suggest and explain what are the precautions that scientists should take to ensure that these are valid. Think about what's going on in this experiment and see what problems could arise that could screw up the changing in temperature and how the respiration rate changes. So imagine we have this fish, and we go from 10, 20, 30, 40, 50, 60, like, okay, 60 bit, like anything above about 40, it's really hot. Well, like, I don't know what temperature fish like, right? But, if you go in a shower and you take a really cold shower, you go like, oh, and start like panicking and like having to breathe, and then you get used to it. So first of all, you'd probably want to, ensure the fish, acclimatizes to the temperature before measuring O2 stuff, right? I think that's just probably most appropriate. And also it avoids temperature, like stress to the fish, like I just said there. As well as that, you probably, again, it's just asking other precautions, the things you control. Other stuff, you might want to measure the amount of salt in the water, and stuff like that, to make sure that the osmotic balance is fine, because it's a saltwater fish. We don't want it to sort of have water moving in and out and all that kind of jazz. As well as that, pH. Why is pH important in this case? You can't just say control pH, you have to say why. So it would be if you had a too high or too low of pH, that can actually affect metabolism of oxygen. Remember that respiration requires enzymes. pH too high, too low can screw up that kind of stuff. And as well as that, we need to make sure this water is sterile as well, because it might have other organisms like bacteria and stuff like that, which could use the oxygen and muck about with your results as well. So those are some of them. So that's what I would say. So ensure the fish blah 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 acclimatizes that, and then you need to say what the precaution actually is. Well, I've just said like allow temperature to stay consistent for a bit for some time before measuring. That way, the fish sets its respiratory rate. Oh, sorry, oxygen respiration levels to the amount that's appropriate for its surroundings. Sorry, my handwriting is getting abysmal. This pen is really, really broken. So you're just gonna have to kind of rely on me speaking. Sorry. The scientists studied the effect of living at different temperatures and the respiration in young fish at different stages of growth. High oxygen consumption is associated with a high growth rate. The results are shown in 20.2. So you got 23 here in the triangles, and it seems to be going down. And as the body mass goes up, the rate of oxygen consumption drops. And then for 18, it's the same, and for 13, it's the same. All of them go down. And then if you compare between them, the rate of oxygen consumption is always consistently higher at 23 degrees versus 18 versus 13, regardless of body mass. So that's interesting. It says calculate the Q10 for the respiration of this, and then give your answer to 3 significant. So the Q10, remember, by definition, is where you're comparing the ratio of two enzyme reactions, or like two reactions, I should say, actually, not just enzymes, two reactions with a 10 °C difference in temperature, right? So based on this, we need to find a rate of oxygen consumption where we have a 10-degree difference. We're not going to do 23 and 18 because that's 5, but we could do 23 and 13 because that's a 10-degree difference, right? And then it says in the largest fish. So we're going to pick the largest fish. So the body mass goes up. So the largest fish would be chilling here. So that means our Q10 is going to be the triangle one of rate of respiration, which is what? Like what's that? 200 over the 13-degree one, which is what? 90? Yeah, 90. Which gives you 2.22. So now it says the scientists said that the best temperature for farming in Cygnus is 18 degrees. So 18 degrees C, best for in Cygnus. And when you're farming it, it's wanting to have like the best growth rates, the highest growth. Right, so it says evaluate the conclusion again, look at their fors and their limitations or against. So if you look at what's for, so it says here, high oxygen consumption in fish is a fast growth associated with a fast growth rate. So if we look at the best temperature for that is 18 degrees C. 18 degrees C is in the middle. You can see that the body mass sort of, as it increases, the oxygen consumption rate drops less. So it means they're growing pretty well. Because here, if you think about it, that's dropping down a bit more steeply. The difference in the two of them is more steep. Sorry, I've drawn that really badly. The difference in the two of them is more steep from top to bottom versus here. So the 18 looks the best. This one's pretty rubbish to start with, and it's also rubbish at the end, like as they get bigger because they're going to grow. So first of all, already we can say that we have a smaller decrease O2 consumption rate at 18 degrees as fish grows compared to 23 and 13. Because if we, as well as that, and I guess another thing you could probably say is that if we're holding them at 23 degrees, it might be expensive. It might not be economic, because that's a pretty specific temperature. Any higher could be too high. So it's a nice middle ground. You could probably say that. The other thing, though, is that some limitations are, well, this only measures the rate of oxygen consumption. We don't just use oxygen for growth, we use it for absolutely everything else. So rate of O2 consumption only suggests, so I should say high rate of O2, suggests increased metabolism, not directly growth. Because remember, like we respire for all kinds of things nowadays, so it doesn't mean that it's 100% growth. As well as that, they're talking about farming fish. We don't want like a really, really high O2 consumption, because that means you've got like high respiration, which means lots of, like food is required. So that's a lot of costs for that. As well as that, they've said it's 18. Well, what if like a temperature between them is even better? So we've only got 3 temperatures used, only 3 temps, no in betweens. I'm being a bit lazy. You would have to say something like there's no, we've only used 3 temperatures. We don't know if there's any higher or lower. Also, as well as that, they've said that farmers should be doing this. Well, I mean, in this experiment, again, line by line, wherever we are, sorry, look at the criticism. Nowhere does it say about like, you know, the sample size. No sample size mentioned. And then, as well as that, we actually have no stats tests either. Because we don't know if these are actually significant differences, or if they did a stats test, there's this massive overlap between all of the results like that, or something. That's useless if there's overlaps. Whereas if they're nice and very defined differences, that's probably a good thing. So now it says glands can either be endocrine or exocrine. So what is an endocrine gland? So first of all, it is cells that secrete hormones. And if it's endocrine, what that means is it's into the blood. So exocrine is more into to sort of like systems like, for example, ducts and stuff like that. And that's more to do with like digestion and things like that. So then it says the pancreas contains endocrine cells, such as islets of Langerhans, and it also has things that produce digestive enzymes. Why are they exocrine? Oh, well, I've just said it there. These are, their substances are released into ducts and not the blood. And I'd be very happy if that's the case, because I do not want like lipase and stuff floating around in my blood. So yeah. Next it says hormones can be classed as either steroid or non-steroid. So steroids are lipid soluble. Suggest the location of the receptors for steroid hormones to suggest a reason for your suggestion. So generally speaking, receptors kind of, you got something that binds to it complementary like that, but they've told you that these are lipid soluble steroids. So if something's a steroid, what it means is if it's lipid soluble, it can move straight through the phospholipid bilayer. So once it's done that and it's crossed into them, remember that like hormones have to actually bind to a receptor and to cause a triggered response. So what it is you've probably got it inside the cytoplasm. Whereas with like a lot of other hormones, they actually can't cross it, which is why the receptors are on the outside there. So based on that, they have to be inside cells, like, in the cytoplasm or whatever. Why? Because steroids can cross the phospholipid bilayer in the cell surface membrane. So now it says the Society for Endocrinology has produced a booklet about adrenal failure to help patients understand their illness. So the following statements are adapted from this booklet. It says adrenal failure is caused by an inability of the adrenal glands to produce sufficient amounts of cortisol and aldosterone. In healthy people, the hormone ACTH is produced by the pituitary gland and causes the adrenal cortex to release more cortisol and aldosterone. A pituitary tumor can stop ACTH production by the pituitary, and this leads to adrenal failure. Symptoms of adrenal failure include severe fatigue and weakness, weight loss, low blood pressure, and salt craving. So explain the symptoms of adrenal failure. So we've got to think straight away what the purpose of all of that information was. So the questions told you it's all to do with the adrenal glands. So the adrenal glands, remember, so if they've told you here, so they've told you it's adrenal glands produce sufficient amounts of cortisol and aldosterone. So they've told you in the question as a reminder that, hello, we're making cortisol and aldosterone. So in the adrenal glands, you've got the adrenal cortex, which is the outer part, and then you've got the medulla as well. So the cortex is what releases hormones that are stimulated from the pituitary gland. So you've got glucocorticoids like cortisol, mineralocorticoids, which is aldosterone, sorry, and androgens like testosterone and stuff. Whereas in the medulla is all to do with when the sympathetic nervous system is stimulated. So they can release catecholamines, and which is a fancy word for saying it, like things like noradrenaline and also adrenaline as well. So in this context, they've said it is adrenal glands making cortisol and aldosterone, and it's to explain the symptoms. So what's happening is there's no cortisol, no aldosterone, and that leads to what? Weakness, weight loss, and so forth. So let's think about why. Cortisol, if there's no cortisol, then what does that do? Well, what does corticosteroids do? You should know that they control metabolism by converting fats and proteins and stuff like that and carbohydrates into energy through your ATP and respiration and so forth. So no cortisol means less carbohydrate metabolism. So if that's the case, then that's going to explain why that they have less ATP and so forth, and they feel tired and things like that. So that explains that one. But what about aldosterone? So no aldosterone. So aldosterone, remember, controls blood pressure and it balances the amount of fluid in your body by controlling how much salt and water retention is going on. So you've got no aldosterone, low blood pressure. So if we've got low blood pressure, you got to think about aldosterone. So remember, aldosterone means more water is retained, more water in your blood vessels, higher blood volume. So what that means is no aldosterone means less water retention. So that decreases blood volume, and therefore you're decreasing your blood pressure. And then in terms of the salt craving part, well, remember, how do we reabsorb water? The general rule of thumb is that sodium ions, where they go, water follows. So if we want to reabsorb water, we're reabsorbing sodium ions. So if we're not absorbing as much water, then that means we've got less Na plus, which gives you in the blood, which gives you the salt cravings. Because remember, in your ascending loop of Henle and all that jazz, what happens is Na plus is pumped out on purpose, that's your DCT, and what that does is it lowers the water potential inside the medulla, so then water can only move out at the descending limb, and so forth. And then it gets reabsorbed into the blood and all of that kind of stuff. So if you've got no aldosterone and it can't do all of that, then you're not taking water into your blood, which means the water volume drops and the blood pressure drops and so forth. So now it says, explain why patients with a pituitary tumor have adrenal failure but still respond normally to situations of danger and excitement. So they've told you what? Let's have a look. So healthy people, so you've got the pituitary gland here, and it makes what? ACTH, and here's your adrenal cortex, and what it does is it makes cortisol and aldosterone. A pituitary tumor can stop it by the pituitary, and that leads to adrenal failure. So if you've got none of that, then they're not making ACTH, and they're not making cortisol and aldosterone. However, it says, explain why these can still have adrenal failure but respond normally to situations of danger. Well, the question has told you that it is the adrenal cortex, and like I said before, the adrenal medulla is still fine. So remember, the adrenal medulla is unaffected, and that's, remember, stimulated by the sympathetic nervous system, which is your fight or flight one, so it can still produce adrenaline and noradrenaline. And the SNS is your fight or flight, and adrenaline is also part of that. So again, it's just ACTH does not affect the adrenal medulla. So now it says hypothyroidism, underactive thyroid gland, is treated by taking thyroxine tablets. Scientists are investigating the use of stem cell transplants to treat hypothyroidism in which thyroid glands of mice are destroyed using radioactive iodine. So the scientists obtained mouse embryonic stem cells and incubated them in cultures with or without a mixture of growth factors for 30 days before transplanting them into the mice. So we've got embryonic stem cells, we've got growth factors, no growth factors, and then we put them in mice. They divide the mice into 4 groups in this table. So thyroid gland destroyed, yes or no, and then we've also got ESCs transplanted in them. So of these, we've got thyroid gland yes, thyroid gland no, thyroid gland yes, thyroid gland no. So just a quick understanding of these things helps a lot. And let's have a quick look at what's going on. So again, compare like for like. So we've got thyroid gland no, and then this no. We've got thyroid gland destroyed yes, this no, and then we've got thyroid glands yes destroyed, but then we've got the stem cells in them, but with or without growth factors, fine. Let's have a quick comparison of what's going on here. So after 8 weeks, they measure the thyroxine concentration of 4 groups of mice. The results are shown in the graph. Each cross represents a single mouse, and the mean plus or minus 2 standard deviations from each group. So group 1, remember, is thyroid gland destroyed and thyroid gland is destroyed, and there is no transplanted stem cells. So they're still making thyroxine, and that makes sense because we've not destroyed their thyroid gland, so that should be working as normal. Next, we've destroyed them and we've not put them in, so that surely should make all of the thyroxine disappear, but you can see there's a little bit still made. Interesting. Next, they have been destroyed with thyroid glands, but now we've given these stem cells, but no growth factors. That makes sense again. The stem cells are there, ready to do its job, but there's nothing to tell them to actually start doing things to make thyroxine, so you'd expect it to be kind of similar. Then we have thyroid glands destroyed, so it should be all the way down here, but then we've given them stem cells with the growth factors, so it should start to crawl back up, which is here. We can see the results are quite spread out there. Fine. Now it says, explain what is meant by pluripotent. It can differentiate or specialize into all adult cell types, or any tissue types, or so forth, or whatever you want to call it. It just cannot form placental tissue, or for example, extraembryonic. Now that I've made sense of the graph, it's quite easy to do this next bit, because all they've said is that ESCs plus growth factors means you get thyroid tissue. So what's for it? Well, you can see that group 4 has higher thyroxine than 3, so that means they can make thyroxine. And they've told you in the question that the thyroid gland makes thyroxine, so surely, based on only these results, that must be the case. And what also supports it is that if you look at group 2 versus 3, you're destroying the thyroid gland, and no growth factors still has low thyroxine. So that suggests that these growth factors are what's making them differentiate and so forth into making the thyroxine. So then, as well as that, 4 nearly makes the same as 1 in terms of thyroxine. However, so a 4 thing here as well is that 1 and 4 results, excluding the anomaly, have overlaps. I'm not talking about the standard deviation bars or anything like that. I'm talking about the actual results. You can see there's 2 up here and there's some there as well, so that's an overlap. So it suggests similar things, thyroxine levels. However, some against is that, 4's mean is lower than 1. Surely if it was working and it differentiated thyroid tissue again, it would be the same. The other thing as well is that their SDs don't overlap. So the standard deviations as lines don't overlap, which means there is a sig difference in thyroxine concentration. You need to say what the difference is that 4 is significantly lower than 1 in terms of its mean. So its mean concentration. So there's no overlap in them, right? The other thing as well is that, if you look at this, we've only got what? So yeah, each cross is a single mass. 1, 2, 3, 4. So again, we're critiquing their experiment. Small sample size. It's a pretty small one because you can see 1, 2, 3, 4, 1, 2, 3, 4. There's 4 per group. So you need to say that. You can't just get away with saying small sample size. You need to actually say to them what's up. And why does that matter? Another thing as well is that 4 has more mice for some reason. Why? So that could affect results. It could, if we did more of these, maybe it'd actually level everything out. And actually maybe get loads more here as well. And as well as that, another point is that thyroxine doesn't directly measure differentiation as well. It's just saying that it's making the hormone. Maybe the hormone comes from somewhere else, and also Surely if you destroyed everything, there'd be no hormones. So there you go. The scientists repeated their experiments with adult stem cells taken from the skin of patients with hypothyroidism. They were artificially transformed into pluripotent stem cells. The scientists concluded the use of human-induced pluripotent stem cells could be safer than using human embryonic stem cells to treat hypothyroidism. Use your knowledge of stem cells to suggest that one reason that supports and one reason that doesn't support this. So number one is that using induced pluripotent stem cells is you're using your own cells, so there's a less chance of rejection, right? Because the immune system, remember, would recognize it as foreign antigens, foreign tissues, and so it would be not good. And bye-bye, like destroyed. The other thing, then, what doesn't support it, however, is that, well, maybe the stem cells they have may have mutations. Because remember, you've got like mitosis over and over and over and over again. Sometimes it's not perfect. So it might have mutations accumulated over time. So that's an issue because it could. So using iPSCs, if you're making them differentiate like crazy, could increase the risk of tumors or cancer as well. Or it might even be that these tissues might still have an allele for hypothyroidism. So even if they are like induced pluripotent stem cells, one of these mutations might actually still stop them from differentiating into it. So it might not be a case of fixing it with stem cells. So that's another thing as well. Now it says, here's a transmission electron micrograph of a chloroplast. Complete the label using the letters from the term that identifies the structure or function. Each letter may be used once, more than once, or not at all. So the chloroplast envelope is that bit on the outside of this whole thing, so that's an F. Site of light-independent reactions. So light-independent, remember, is in your stroma, so that's in the juicy bits there. E. Intergranal lamellae. So remember, those are what join those stacked thylakoids. So that's going to be, so you got your stacks here and they're linked together like that. That's D. And then contains starch grains and lipid droplets. Again, it's the stroma, so it's E. So now it says complete the sentences using the most appropriate terms. So the carbon fixation occurs in the Calvin cycle when CO2 combines with a 5-carbon compound called ribulose bisphosphate. This forms, you can't really just say, I'd avoid saying RUBP and just say the whole thing, and then if you want to say RUBP, bracket it later. This forms an unstable 6-carbon compound that immediately breaks down into 2 molecules of glycerate 3-phosphate. These are reduced to triose phosphate using reduced NADP and ATP that are formed in the light-dependent stage of photosynthesis. Triose phosphate is then used to make hexose phosphates, which can be converted to something for transport elsewhere on the plant. It's sucrose. So the whole point of the light-dependent reaction is to generate reduced NADP or NADPH or whatever you want to call it, and ATP, so that this can happen. Remember that the end goal is you want to make three carbon TPs, and you start with a 5-carbon RUBP, which is 5C. In order to get to 3, we need to, 5 and 3 aren't nice numbers, so what we do is we stick a CO2 in it using Rubisco, and that gives you that 6-carbon compound, which becomes 3 times GP. Now, again, all the way up to now, we haven't actually used any of the light-dependent reaction stuff, so it must be at this stage where that kind of stuff happens, where your reduced NADP becomes NADP. So the GP has been reduced, and this has been oxidized. This reduced NADP has been oxidized back into NAD, and in order to do that, it requires energy. So ATP becomes ADP and PI. So it's just really important to understand the interactions between each of those reactions as well. Sorry that was really rushed. My pen is on its last legs, so I'm just trying to get through these as much as I can before in the time I've got, getting a replacement. So thank you very much. I hope that helps. Please like, share, and subscribe. You know the drill.